So picture a grain of sand,
Right.
just resting on your fingertip.
Okay, pretty tiny.
Right, tiny. Now imagine that inside that single tiny grain, there are nearly 150 million separate biological wires.
Wow.
And they're actively firing electrical signals back and forth in this network that is just so dense, it completely defies human comprehension.
It really does.
But that is exactly what a cubic millimeter of your brain actually looks like. So welcome to this custom-tailored deep dive. If you're a curious mind looking for those, real aha moments without drowning in a textbook, you are in the perfect place.
Absolutely.
Today, our mission is to explore what I'd call the ultimate frontier. We are talking about mapping the brain at the resolution of a single synapse. And we want to answer that massive, long-standing question. Can a computer actually simulate brain activity?
Right, which is the holy grail of neuroscience, honestly.
Yeah, exactly. So we're pulling from Google Research's H01 dataset today, which is this mind-bending reconstruction of a millimeter of the human temporal cortex. And we're also looking at some fascinating studies on sensory-motor processing in model organisms.
Yeah, the Google data set is just, the scale of it is unprecedented.
It is. To anchor your imagination here, in just 1 cubic millimeter of human brain tissue, researchers painstakingly mapped exactly 149,871,669 synapses.
Which is just a staggering number to even say out loud.
It is. And the breakdown is wild, too. It's roughly 111.6 million excitatory synapses and 70.7 million inhibitory ones. And they did this all using an interactive 3D web tool called Neuroglancer.
Yeah, Neuroglancer is amazing because you can actually, you can boot it up in a browser and literally fly through the tissue.
Right. I spent some time doing exactly that. And the first major aha moment for me had absolutely nothing to do with neurons.
Which is weird because when we think of the brain, we immediately picture neurons, right?
Sure, the thinking cells. Exactly. But looking at the raw data for this temporal cortex sample, the non-neuronal cells, the glia, they actually outnumber the neurons 2 to 1.
Yeah, it's a massive ratio.
Like there are 32,315 glial cells packed in there compared to just 16,087 neurons. The neuron density in this human sample is actually 10 times lower than what you'd find in like the association cortex of a mouse.
Which really forces us to reevaluate what all those support cells are actually doing.
Yeah.
Because for decades, science kind of treated glia as essentially biological packing peanuts.
Packing peanuts.
Yeah, like the assumption was they just fill the empty space, provided basic metabolic support and just kept the important cells alive. But this data set proves they have a highly specific, rigid spatial organization.
Okay, how specific are we talking?
Well, when you look at microglia and oligodendrocyte precursor cells, or OPCs, they show this massive affinity for blood vessels. A huge portion of them resides strictly within 5 micrometers of the vasculature.
I mean, that makes sense on a surface level, right? They need nutrients, so they hug the blood vessels.
You'd think so. But then you look at the white matter. The oligodendrocytes there form these distinct lines along radially oriented blood vessels. And crucially, they cluster specifically above and below the large blood vessels.
Wait. So they completely avoid the sides?
Exactly. They sit on the top and bottom, but not the sides. And the researchers concluded this isn't for metabolic reasons at all.
Oh, really?
No, it's entirely mechanical. It's due to a physical constraint called tissue anisotropy.
Tissue anisotropy. Okay, what does that actually mean in this context?
So in this specific region, the vast majority of the myelinated axons are running strictly along a single axis. The dataset calls it the z-axis.
Okay, so millions of microscopic cables all running in the same direction.
Right. And because they're packed so tightly, they act like rigid structural pillars.
Hmm.
This physically, literally forces the glial cells into specific vertical positions. The physical environment dictates that there is simply no room on the sides.
Okay, let's unpack this because that is wild. It's like commuters on a crowded subway car, right?
Yeah, that's a good way to look at it.
Like, you are being physically forced to stand on specific sides of a pole, not because you want to, but because of the sheer crush of people moving in one direction.
Exactly. The architecture of the brain is constrained by its own structural mass.
So if the physical environment mechanically forces the support staff into these rigid structural patterns, it begs the question, are the neurons themselves subject to strict, bizarre geometric rules?
Oh, they definitely are. And this brings us to one of the most mysterious findings in the whole data set.
The compass neurons.
Yes. So moving from glia to neurons, the researchers looked deep in the cortex, specifically layers 5 and 6. And they found this highly structured subcategory of cells. They call them triangular or compass neurons.
Right, I saw those in the 3D models. Because a normal pyramidal neuron has like a skirt of dendrites reaching out everywhere, but these compass neurons have one uniquely large basal
A
dendrite.
massive dominant dendrite, yeah. But the real discovery is where that dendrite points.
All right.
They plotted the coordinates. The theta angle, which measures the radial projection, averages about 126 degrees, pointing down toward the white matter.
Okay.
But then they mapped the phi angle, the angular coordinate, and they found a strict bimodal distribution.
Meaning they point in two mirror-symmetrical directions, right? Along the anterior-posterior axis of the temporal lobe.
Exactly. You have this reverse-going group pointing toward the beginning of the tissue sac, with a phi peak at exactly 90 degrees.
Wow.
And a forward-going group pointing to the end of the stack with a phi peak at 270 degrees.
That is so specific.
It is. There's also a smaller tangential group sitting between like 0 to 45 degrees and 135 to 225 degrees, but the main groups are locked at 90 and 270.
So what does this all mean? Like, what is the biological function of having neurons act like biological compass needles?
Well, the fascinating anticlimax here is that science currently has absolutely no idea.
Right, really? None at all.
I mean, there are hypotheses, but no definitive answer. They show significant anatomical clustering, meaning they point the same way as their neighbors. But their actual purpose remains one of the dataset's greatest mysteries.
That is incredible. We see that the physical shapes and orientations of neurons are highly organized, but, structure is really only half the battle.
Right. It's about how they communicate.
Exactly. How do they talk across these structures? Which brings us to the actual wiring and the discovery of these rare, hyper-powerful connections. Because when I was looking at how synapses actually connect, it seems like most of it is just random.
Yeah, the vast majority follows Peter's rule. About 96.49% of axonal inputs are just weak single synapse connections.
So an axon just happens to cross a dendrite, and a connection forms purely by accident.
Right, just an accident of proximity. But then they found these anomalies, rare, incredibly powerful connections where you have up to 53 synapses from a single axon to a single target cell.
53 synapses, from one axon to one cell.
Yes, and it happens through something called a directed growth motif.
Okay, how does that work?
So instead of cofasciculation, where the axon just travels alongside the dendrite, the axon hits the dendrite, forms a synapse, and instead of moving on, it radically alters its behavior.
It doesn't just keep growing past it.
No, it sprouts multiple terminal branches up and down to aggressively grab that exact same cell over and over again.
Okay, here's where it gets really interesting. Because instead of high-fiving someone in passing, this is like an axon stopping in its tracks, growing 10 extra arms, and aggressively shaking that one specific dendrite's hand 50 times.
That is exactly what it's doing.
But wait, how do we know this isn't just a random accident of growth? In a dense space, things could just get tangled, right?
Right, and researchers wanted to prove intentionality, so they built a brilliant null model.
A null model, like a computer simulation.
Exactly. They simulated randomly displacing the mapped axons by 15 micrometers.
Oh, so they just shifted them over slightly in the virtual space.
Yes, and then they let them form the same number of synapses based on strict distance parameters.
Like what kind of parameters?
Like, en passant, synapses were allowed to form at 270 nanometers and stock-type synapses at 1500 nanometers.
Okay, so sticking to biological rules, but shuffling the deck.
Right, and the result? The chance of these massive connections happening randomly is practically zero.
Wow.
The p-value was less than 10 to the negative 10th. It proves the brain is intentionally prioritizing specific connections.
So we now know the brain intentionally creates highly specific, powerful circuits. But mapping a circuit is just looking at a static map, right?
It is. It's anatomy, not action.
Exactly. To understand how a map turns into an action like, human intelligence or physical behavior, we have to look at how these circuits process sensory input.
And to do that, we move from the human temporal lobe to model organisms, where we actually have the complete wiring diagram.
Right, organisms like Drosophila, the fruit fly, and C. elegans, the roundworm. This is what we call sensor motor processing.
Yeah.
And the fruit fly example is just fascinating.
Oh, it's amazing.
Like, if a fly tastes sugar on one leg, it doesn't just stick its proboscis out randomly hoping to find food. It actually extends it directionally towards the food.
Right, and because we have the connectome, we can trace exactly how that happens. They use computational models specifically, leaky integrate and fire models, or LFF models.
Okay, leaky integrate and fire. Break that down for me.
Sure. Think of a neuron like a leaky bucket. As signals come in, voltage fills the bucket. but it naturally leaks over time.
Yeah.
If the signals arrive fast enough and strong enough, the bucket fills up and the neuron fires. Using these LF models, researchers achieved 91% consistency in predicting the fly's actual behaviors.
91%, just from simulating the physical wiring map.
Yes. They can see how sugar activates gustatory receptor neurons, which then trigger specific motor neurons like MN9, which literally controls the rostrum lifting.
That is mind-blowing. And we see this in other behaviors too, right? Like antennal grooming.
Antennal grooming, yes.
Yeah.
And ocellular flight control.
Right, the flight control, where they use 3 small dorsal eyes to detect light shifts. And the circuit instantly balances excitation and inhibition for flight stabilization.
Exactly. The map predicts the exact behavior.
And what really stood out to me was how different sensory modalities move at different speeds. Like, visual and mechanosensory signals reach the motor targets at a much earlier rank or layer in the network than other senses.
Right. The physical architecture prioritizes speed for crucial survival inputs. But, and this is big, but there are vital caveats here.
Oh, for sure. The map isn't everything.
No, we can map the anatomy perfectly, but things like neuromodulation completely alter the rules.
Neuromodulation, like the chemical soup.
Exactly. Chemical signals, like neuropeptides, can wash over a brain region and change how the whole circuit behaves without changing the physical wires. Plus, current models struggle with a basal inhibition.
Right, the baseline level of suppression that's always kind of running in the background.
Yeah. And beyond the chemistry, you also need to combine the neural model with a biomechanical model of the physical body to truly simulate behavior.
Which is what the OpenWorm project is doing, right?
Yes. They take the C302 neural model of the roundworm and combine it with the cybernetic biomechanical body.
They basically plug the digital brain into a physics engine to see if it swims.
And it does.
Hmm.
But it shows that simulating life requires both the wiring and the physical constraints of the body.
Man, okay. Well, let's just recap this journey because we have covered so much ground today.
It's a lot to take in.
We started with 150 million synapses in a grain of sand, and the mechanical rules, the tissue anisotropy dictating where glia can physically exist.
Right.
Then we discovered those mysterious compass neurons locked at exactly 90 and 270 degrees.
It's still a mystery.
Still a mystery. Then we proved the brain intentionally builds hyperpowerful multisynaptic connections using that 15 micrometer null model displacement. And finally, we saw how mapping these circuits allows computers to predict physical behaviors in a fruit fly with 91% accuracy using those leaky integrate and fire models.
It's an incredible chain of discovery.
And for you, listening, this is why this matters. Understanding these sources proves that simulating brain activity isn't just a sci-fi concept anymore. It's actively happening right now by combining massive anatomical mapping with computational models.
It is. But I think it leaves us with a really provocative final question.
Oh, lay it on us.
Well, if the physical architecture of the brain is so deeply constrained by mechanical forces like tissue anisotropy forcing glia into place, or specific neurons pointing to strict 90 and 270 degree coordinates, how much of our flexible, unpredictable internal state is driven by that rigid physical wiring?
Oh, wow.
And how much is just floating in the invisible chemical soup of neuromodulation that we literally can't see on an anatomical map?
That is going to keep me up at night. Seriously, the machinery inside your own head is just unimaginably complex. Thank you so much for joining us on this deep dive today. Keep exploring, keep questioning, and we'll catch you next time.